Good afternoon, everyone. I'm Peter Johnson from Medical Research at Avery, Dennison. I'd like to ask you all to stand to sing the national anthem, but I realize that that's a high-risk maneuver to begin a talk like this these days. Here to talk to you about temporary adherent medical devices and new approaches to their management. Temporary adherent medical devices are things we use every day. We're here at this great convention looking at a litany of medical devices that don't fit into this category, and we're actually hearing some being made in the background. But temporary adherent medical devices are very important to all of us. They tend to be the things we use day to day in our homes, such as band-aids. They're also surgical incise drapes, wearable sensors, ostomy appliances, things that are vital for us day to day that we need to find the best technologies to put against our body so that they form what is really, in fact, the most intimate interface with our bodies of anything that's applied to us externally for any great length of time. So we pay a lot of attention to these at Avery Dennison, primarily through the development of the adhesives and the films that are needed to make these into strong products. Pressure-sensitive adhesives are the basis for the attachment of temporary adherent medical devices to the human body. A pressure-sensitive adhesive has three components to it. One is the substrate to which it's being attached. In this case, generally, it is intact skin or skin around a wounded area. The pressure-sensitive adhesive itself, which is a chemical formulation that exhibits both cohesion, the ability of molecules to stick together with one another almost as a team, and adhesion, the ability of those molecules to interact with the topography of the tissue and the surface energy of the tissue and to wet out or to find the best possible way of making the highest level attachment to that interface. And then finally, a backing film that's designed carefully so that when it's applied, it holds the adhesive in place with a structure, but also allows for removal that's as minimally traumatic as it can possibly be. What is the range of skin adhesives that we see? Well, rubber-based adhesives have very high capability of adhering to skin. You'll notice that we have two axes here. One is the MVTR FHC axis, which is the moisture vapor transmission rate and fluid handling capacity. And you'll notice that when the moisture vapor transmission rate, that is the ability of sweat and other fluids to transmit through the adhesive barrier, is low, it tends to be characterized by rubber adhesives and acrylic adhesives, but it's much higher transmission rates when water-based materials such as hydrogels are used, which are essentially emulsions that have adhesive characteristics and also hold moisture into the wound itself. Silicone adhesive and polyurethane adhesives have middle ground properties. These tend to be today's most atraumatic adhesives, but things are changing as acrylic adhesives are modified so they retain this high tension of adhesion, but also better and better moisture vapor transmission rates and easier removability. There are some complex considerations when you're dealing with skin. None of us in this room have skin that's all the same over our entire bodies. As a matter of fact, our skin changes across our lifespan, so that what we were once when we were young children is completely different by the time we're in middle age or adults. If you begin to look across the spectrum of just the people in this room, the range of skin qualities varies dramatically. And if you look across the entire world, of course, you're looking at a spectrum of human characteristics that is quite different and very difficult to characterize using present technologies. So this intra and inter-individual variability is something that we're trying to attack at Avery Dennison so we can begin to identify adhesives and film combinations that are personalized for groups of people that are optimally adhesive, that deliver the appropriate characteristics we want, but can be removed without tearing skin or otherwise damaging the tissues that lie beneath the skin. You notice that the skin is actually a second kidney. Your kidney secretes something less than 2,000 cc's of fluid per day. Your skin eliminates 600 milliliters of water per day. What's very interesting about your skin is that on the outside of your forearm, for example, you might lose 2 to 8 grams per meter squared per hour of tissue of that type. But on your hands and your armpits and the soles of your feet, you might lose up to 10 or 15 times as much water. And if you damage the skin, primarily through abrasion, if you have an abrasion wound and you've got relatively intact skin, but where the lining of the skin, the epithelium, has been lost in certain parts, you can lose a dramatic amount of water, almost equivalent to what the normal overall water loss is per day. But the skin isn't just a water loss mechanism. The skin also has a way of exuding lipids through its surface, which is why we have oily skin. It's essentially a product which, when found in variations around the body, impede the ability of pressure-sensitive adhesives to wet out on those surfaces. So some attention needs to be paid to those parts of the body that have oilier characteristics to find adhesives that can actually work there reliably, not only for bandages, but also for things like the guidance of surgical systems that require close fixation of adhesives to the tissue, for example, on foreheads, which are typically quite oily. And then finally, what's known as the stratum corneum is just simply the top lining of the epithelium of the skin. It's the part that is the best moisture barrier of the skin. It's where the skin cells actually fill with protein and die, forming a barrier. And this area has what's called low surface energy and pH that's less than 7. And for those of you who took high school chemistry like I did, that means it's slightly acidic. And so adhesives need to be built so that they take into consideration not only the surface energy, but also the acidity of the skin. What we've done at Avery is to recognize that up until this time, the laboratory rat of the adhesion world has been what's called the volar forearm, or the undersurface of the forearm. Almost all adhesion testing that's done on human beings is done on this one part of the body, which is it's done there typically because it's relatively hairless, except in some unlucky men. And it can be typically made reproducible on a single individual. But remember, that part of the body is very rarely wounded. Most bandages are placed on the surface of the knee, or on the soles of the feet, or on elbows, or places where there's a tremendous amount of energy being transmitted through the adhesive and the film. And the testing of adhesives is being done in a relatively static area, chosen primarily because they don't want the tested population to feel pain when these adhesives are being removed. We're taking an entirely different approach at Avery. We're looking at testing adhesives all over the body and looking at the most difficult places where adhesives are typically used. These are just diagrammatic activities, but what we've done in the company is to build a research review mechanism that allows us to do fairly rapid sampling of subpopulations to obtain data that can be designed to be indicative of an individual's variability, but also the variability between individuals. Now, the way that these are tested in these different regions is through the application of a number of different tests, not all of which I'm going to read to you here, some of which are applied before an adhesive is used in order to characterize the tissue, some of which are applied after an adhesive is taken away in order to determine what happened to the tissue as a result of the adhesive being there. For example, some of them are moisture vapor transmission, others are topography of the skin, others are indices of inflammation, such as the color of the skin before and after, and sensitivity testing on skin. But in addition to that, an adhesive is actually a biopsy instrument. When you put an adhesive on an individual, you can never take one away without removing at least some of the cells on the surface of the epithelium of the skin. That turns into a very interesting opportunity to study those biologically and actually create a systems biology analysis that allows you to personalize the way that an individual can react to an adhesive. Through the study of DNA, RNA, various metabolites of the skin, we can begin to determine what correlates with the reactions we're seeing on a more macroscopic level. The most important thing we look at when we're looking at adhesives on the human body, though, is pain. If you ask nurses, what is it they rue the most about the adhesives that they apply to their patients? It's in particularly young children and particularly premature infants who, if they have skin torn at the time of removal, can sometimes die of sepsis from the wound that's created. They say the most worrisome thing to them is the pain that they have to inflict in order to take these adhesives away. Under many circumstances, such as negative pressure wound therapy, where a large wound is covered with an adhesive drape and suction is applied to augment the ability of the wound to contract and to create new cell populations to close the wound, those adhesives need to be so strong to be able to tolerate the vacuum and the pulling on the tissue combined that when they're removed, they're often the most painful of dressings to remove. We're looking at new ways to make those atraumatic as well. In essence, what we've created is a system at Avery where what we can begin with is a comprehensive skin testing mechanism that not only tests adhesives on multiple sites in the body that are relevant for the application of those adhesives, across an individual and across populations of individuals, aggregate that data to identify commonalities and begin to identify essentially response biomarkers to the application of adhesives that allow us to design adhesives in a better way for different populations of individuals. The customized testing array that's done is followed by a data analysis, which is a way of providing correlative bioinformatics across a spectrum from the tissue changes that you see macroscopically, the psychological response to the adhesive, and also the biological reactivity of the components that are removed with the adhesives themselves. These are then put into our system at Avery to do creative development of adhesive film and film combinations so that we can optimize adhesives. Not to say that every individual is going to have their own adhesive pattern, but it's very likely that what will happen and will fall out is almost histogram-like patterns of preferred adhesives for different types of individuals that may even change over their lifespans. And that's what we're really looking forward to putting forth in the years to come. At Avery, we have core technology platforms that allow us to do this. We have a complete range of skin contact adhesives, including hydrocolloids, can incorporate drugs into adhesives such as antimicrobials, and also incorporate polymers such as hydrogels that are much more cell-friendly and protein-friendly at the interfaces, particularly with wounds. We also can incorporate odor-absorbing technology, particularly for wounds and for ostomy appliances. And then finally, films and foams and other products that go on top of the adhesives can make their characteristics optimal for the specific sites and the specific individuals, particularly with respect to the pain scale that we talked about. At Avery, there's a commitment to R&D that crosses a broad spectrum. There's a core capability at Avery called the Avery Research Center, located in Pasadena, where 80+ scientists are engaged in materials characterization, new technology development, and interfacing with the various divisions at Avery that are building out these scaled-up technologies. So we have a core R&D function that we can draw upon when we identify the data sets that direct us to the building of specific types of products for different populations. We also have in Mill Hall, Pennsylvania, the Performance Polymers Division that's focused on the development of adhesives and other polymers using state-of-the-art equipment and state-of-the-art talent, so that as they react to what we identify as the sensitivity patterns, for example, of children, they then can build adhesives specifically for them. Or older people who are on steroids for a transplantation with very thin skin, they can identify polymers that work optimally for them. And in correlation with the work being done at the Avery Research Center, they can make these even less traumatic using new physical mechanisms. Active ingredient delivery is something that we're engaged in today, primarily today in the area of antimicrobials for multiple applications, from IV securement out through ostomy. But as you can see, there are multiple ways in which our technologies can be used to incorporate various active ingredients to be delivered to wounds. And we won't go into some of the more futuristic applications, but I can indicate to you that we're thinking about everything from along this list and more to be able to build better and better products as we go forward. I mentioned earlier in this talk that the films that are used as backing for adhesives are just as important as the adhesives themselves, because think about it. If you've got a bandage on that's got a very thin film and you're trying to remove it, that angle that it draws your skin can be more painful than perhaps a thicker film that allows you to break it away with a little bit more clarity. What we do is we integrate the knowledge that we generate from our data analysis with what we learn from the Polymer Center, put on the proper films, and then our Film Center has the capacity for cast extrusion, identification of multiple vendors that can bring the appropriate materials in, and also even then go on to do blown film processing when necessary. The adhesive coating capabilities at Avery are such that using computational analytical techniques can be brought down to within 1 micron of the coat width that's needed. So the optimization of product can be made not only from the adhesive to the film, but also to the combination of the two at the thickness level for the different populations that we've discussed. Very interesting technology that we apply, particularly in the area of sensors, and also in a couple of other applications, particularly negative pressure wound therapy, is multi-laminate manufacturing, where web-to-web based roll manufacturing is used to create polylaminates that include adhesives and films that are up to 10 to 15 layers thick, reliably and at high speed. In the end, what we're looking to do is to advance our own intelligence so that we can optimize the patient temporary adhesive medical device interface. To review, we've established a multi-site skin adhesion testing system that's under internal research review, a full range of skin assays, data analytics that allow for correlative bioinformatics across the spectrum, from not only the wear testing but to the biological response. data-driven adhesive film development, including additives, and then finally the full manufacturing capabilities so that we can finally look at adhesives today not as something where one size fits all, but where we can begin thinking about all of us as unique individuals that require adhesives that are designed for the needs that we know are going to be most important for us and for our families. And with that, I'd like to stop and take any questions you might have. Can you use the microphone?
Are you actually manufacturing hydrogels at this point, or you just have the capabilities?
We're manufacturing polymer hydrogels. It's polyhema hydrogel.
Okay.
So I think one of the interesting things you mentioned was the customizability or tuning of various adhesives to different skin types. In terms of implementation of that, that certainly would be a question from a medical device manufacturer side, such as my company. We'd like to be able to tune adhesives for patients, obviously. However, practically speaking, have you guys thought about how you might actually implement that? Because that clearly would lead to potentially needing many more different types of dressings if you really need to tune it to that degree.
Right, and that's where we need to distill the complexity into meaningful subsets that make business sense. Clearly, when we talk about personalized adhesives, we probably are not going to get to the point where we have 3 billion types of adhesives, but we probably will have the ability to parse what we have into meaningful subgroups that are of lower trauma, longer duration, and less potential for bioincompatibility.
And is there a timeline you have associated with trying
no particular timeline. This is an ongoing effort for us.
Is your company involved with either Tegaderm or with wound vac products?
We're very heavily involved in the latter, and in the former is not our product. We have similar products, but that's not our product.
Can you explain how do you test the biocompatibility? The second question is do you need FDA approval on your product?
Yeah, okay, so if we brand our products and we make them our own medical devices, we will need FDA approval, and we need to live within a design control-based quality system. And we have that in place, and actually emerging, we have that in place in our European facility, and we're now having it in place in our US facility by the end of this year. With respect to the testing of biocompatibility, we typically do that with an outside source, and we follow the ISO 10993 process. So there's sensitization, irritation, and cytotoxicity are the 3 things that are required for a non-invasive based skin contact adhesive. Hope that answers your question. Well, thank you very much, everyone.